Good morning. In my previous video, I have talked about the discovery of various types of plant growth regulators under the chapter Plant Growth and Development. This video onwards, I would like to discuss site of synthesis, function and the physiological effects of various types of plant growth regulators or the phytohormones. First, we will start with auxin. Let's begin. Auxins are the derivatives of indole compounds, and the structure of the chemical structure of the auxin is like this. Auxins are the plant growth regulators which are generally produced at the tip of the stem and the root, from where they are transported to other parts of the plants. This promotes various types of functions, but before that, I would like to tell you that the auxins are of two types. First one is the natural auxin, the auxins which are produced naturally within the plants. Example, indole 3 acetic acid, indole butyric acid, etc. And the synthetic auxin, that means the auxins are prepared, synthesized artificially for the benefit of the mankind and used in the agricultural field. Example, naphthalene acetic acid, 2,4-dichlorophenoxy acetic acid, 2,4,5-trichlorophenoxy acetic acid. Now let's start to discuss about the different types of functions that are carried out by auxins. Auxin promotes apical dominance. The term apical dominance is very important. What does it mean? Apical bud contains apical meristem. In apical bud, the concentration of auxin gets increased, thus responsible for increasing the rate of cell division of the apical meristem and thus helps in stem elongation, increase in height. And the lateral buds are present in the axils of the branches. This high concentration of auxin in the apical bud inhibits the growth of the lateral bud, and high concentration of auxin promotes the growth of the apical bud. That is called apical dominance. Auxin does not let the lateral bud to grow. It prevents the development of the lateral bud as long as apical bud is dominant and keep secreting auxins. Auxin promotes cell division. Auxin involves in the initiation of the root and stem cutting. That is, in vegetative propagation, the stems are cut and then dipped in the auxin solution and thus helps in the initiation of root. And also it helps in lateral branching of root, and for this indole acetic acid, naphthalene acetic acid, indole butyric acids are used. The next one is the tropical movement. Phototropic and the geotropic movements of the plants are regulated by differential concentration of the auxin. Auxin helps bending of the stem towards the light, and the root moves away from the light towards the force of gravity, that is called the geotropism. It will be easy if we see this animation. This is sun, the site of the stem which is nearer to the light. here the auxin concentration gets increased. This auxin then diffuses to the darker region and responsible for increasing the rate of cell division. Auxins are synthesized in the lighter region, diffuses to the darker region, responsible for increasing the rate of cell division, and here the rate of cell division is less. Differential cell division helps in bending of the stem towards the light, that is the phototropism. Auxins work differently on leaf fall and the fruit fall at different time periods. It prevents premature leaf fall or the fruit fall. It promotes abscission, that is the detachment of the old leaves and the matured fruits or the ripened fruits. Auxin induces parthenocarpy, that is the production of the fruits without fertilization. High concentration of auxin induces fruit formation and hence the fruits that are produced that are seedless. Example, tomato, banana are produced in this way. Naphthalene acetic acid, indole butyric acids are used. Auxin also helps to regulate xylem differentiation. Auxins are widely used as herbicides to kill the dicotyledonous weeds in the garden or in the lawn. In the lawn, the grasses are grown. Grasses do not affected by auxin, but the dicotyledonous, broad-leaved, unwanted weedicides or the herbicides are killed by the high concentration of auxin like 2,4-D, 2,4,5-3, etc. This high concentration of auxin actually responsible for abnormal cell division in the root and thus responsible for blocking the xylem and phloem, thus hampers conduction of the nutrients and the water and ultimately responsible for killing the death of the dicotyledonous weeds. So, to make the lawn free from the weeds, gardeners use auxin, 2,4-D, 2,4,5-trichlorophenoxyacetic acid, this type of synthetic auxins. Auxin helps in plant propagation. Auxin promotes cell division. Auxin helps in root initiation and the branching of the root. All these features, the functions are used in the agricultural field for the vegetative propagation of the plants. High concentration of auxin induces parthenocarpy, that is, the fruits are produced without fertilization, thus the fruits are seedless. That already I discussed. So the banana, tomato are commercially produced by increasing the auxin concentration. In tissue culture, auxin and cytokinin are used when the auxin concentration gets increased. That high concentration of auxin compared to cytokinin helps in root formation. Now some more informations about auxins. Some auxins cannot be easily extracted or isolated from the plants. These auxins are called bound auxins. For the extraction of these auxin, organic solvents are required. Some auxins can be easily extracted or isolated from the plants. These type of auxins are called free auxins. Now another important terminology, that is antioxin. The compounds which inhibits the activity of the auxin are called antioxin. Example, p-chlorophenoxyisobutyric acid, PCIB. Now in this video, I have discussed the site of synthesis of auxin and also the functions and the physiological effects of auxin and how these functions can be utilized in agriculture and the tissue culture technique, that also we have discussed. I hope it is clear to you, my dear child.